Topic: Phytohormone regulation of plant growth and development. Number 1. Key issues. Topic plan. First, hormone regulation of ontogenetic problems. Second, embryogenesis. Third, plant meristems. The purpose of the lecture: the study of phytohormone regulation of plant growth and development.
And angiosperms are the flowering plants, and they can be divided into two different categories, the monocots and the dicots. Monocots have one seed leaf, one cotyledon in the seed, and dicots, meaning two, have two seed leaves. Now you don't have to just look at the seeds in order to be able to make some comparisons. Monocots have obviously only one cotyledon, but they also have veins in their leaves that are parallel in arrangements. They have floral parts that are usually in multiples of 3s, and some examples would include corn, wheat, lilies, orchids, and palms. Dicots, on the other hand, obviously have two cotyledons. Their veins are usually more net-like. They have vascular bundle that's arranged in a ring, and they have floral parts that are in multiples of 4 or 5s. Some good examples would include roses, clover, tomatoes, oaks, daisies, and a number of other different species. Plants mostly experience something called intermediate growth, and the lifespan of a plant is genetically determined, meaning it's based off of the genes, obviously. Annuals complete their life cycle in a single year. Some examples would be like marigolds, cucumbers, lots of garden plants. These would be things that you would plant on an annual basis. Biennials complete their life cycle in 2 years, and so in the first year they're germinating and growing the roots, and in the second year they're growing stems and leaves, and they produce flowers and seeds. Some examples would include primrose and celery. Perennials are plants that would live for more than 2 years, and this is often like some of the bigger trees, would include like maple trees, palm trees, and various grasses. The overall structure of the plant is it's made up primarily of a root and shoot system. So here's the root system, and then here's the shoot system. And obviously there's leaves and different nodes and sometimes flowers and branches that are included in the shoot system. The tissue in the stem and leaf has some unique features, and some of these we've already looked at. We've looked at the vascular bundle, which includes the xylem. The cambium is a small section that separates the xylem from the phloem. The epidermis would be the outer cells. The pith would be inside portion, and then the cortex. Here's a different cross-section of a leaf in dicot specifically. And we have an upper epidermis, the palisade mesophyll, where a lot of those cells more specific for photosynthesis would be found. The xylem, phloem, cambium, part of the vascular bundle. And then the lower portion of the leaf is going to have the spongy mesophyll. It's going to have a lot of space that allows for gases, CO2 and oxygen to be able to move and diffuse through. And then we have a lower epidermis with stomata. Above and below on the upper and lower epidermis, you could also find a waxy cuticle, and that helps to retain water, to actually keep water in. If we look at the structure in a little bit more detail, the stomata and the guard cells, gas needs to be able to get in and out of the plant leaves. And so the diffusion of these gases occurs through stomata. And there are small openings inside of, or on the bottom portion of the leaf. And they're controlled by something called guard cells, which will close to help retain water, which would obviously then block the diffusion of gases, and it can stop or reduce the rate of transpiration. The epidermis layer of the leaf tissue is for support and water conservation. The outer layer protects the inner mesophyll layers. It's got a thick wall for strength and support. It's transparent to allow for light to be absorbed. Obviously, that's very important in plants. And it secretes a lipid layer, which creates that waxy cuticle that helps to prevent water loss. The vascular bundle, as we've talked about, transport water and photosynthetic products, sugar throughout the plant. The xylem transports the water. The phloem is what's primarily transporting those sugars. So now we want to get into some of the more specific topic for this section 9.3. We want to look at undifferentiated cells and mitosis. And totipotent cells, many plant cells including fully differentiated plant cells, have the capacity to generate whole parts. And where the plant is growing typically is in a root meristem or apical meristem. And we'll look at those individually here in just a second. Here's the apical meristem, and it kind of looks like some weird alien thing, but this is where the plant is growing vertically. So these would be some auxiliary buds that would then produce additional shoots, but the plant is growing vertically from this apical meristem. And the growth is going to be confined to these regions called the meristems. And those are regions that are composed of undifferentiated cells that are undergoing active cell division. So these cells are dividing by cell division, and that's causing the plant to grow in these different regions. So let's take a look at growth in apical meristems. These are found at the tips of the stems and the roots, and they're primarily responsible for growth. And they increase in length of these different systems, again causing primary growth. It occurs by cell divisions, and this is occurring by mitosis. And so these are generally small cells that go through the cell cycle of mitosis to make more cells multiple times repeatedly in order to create more and more new cells. And so how this works in the shoot apical meristem is it's producing stem and cells that develop into leaves and flowers. And how this works is that with each cell division, one cell remains in the meristem, while the others increase in size and eventually then differentiate due to some different gene expressions to form the meristem region, and that will eventually go on to perform or to become the different leaf structure, the stem structure, or whatever it may be based off of what the genes are telling it to do. And so one cell is remaining that continues to divide, and that remains in the meristem while the others are differentiated to become whatever structure the plant needs. And so the apical meristem overall gives rise or produces the epidermis, ground tissue, the vascular tissue, including the xylem and the phloem, as well as the pith. And this all produces initial tissue in the growing plants. The lateral meristems are what increases the lateral or secondary growth. And so this is how the plant gets wider, where the circumference and strength as well as the stem increases. And this occurs at the cambium, which is between the xylem and the phloem, and it forms a secondary xylem or phloem. And this occurs more actively in older stems and root tissue. So now that we've seen and examined how plants can grow vertically and increase in circumference and their width, let's talk about some different plant hormones that can influence the shoot growth. And auxins is a type of hormone that is responsible for this. And a hormone is some sort of chemical message that's produced and released in one part of an organism that affects another part of an organism. And there's lots of different hormones, including those in the human body. There's one in particular in plants called auxin indole 3-acetic acid. So it's a type of auxin. And it controls the growth of the shoot apex. And it promotes the elongation of cells in the stems. Can also can inhibit growth in high amounts. And so it can also, different auxins can initiate root and fruits and leaf development and growth, and this one in particular controls the growth in the shoot apex. Nodes are regions of the meristem that are left behind as the shoot apical meristem grows. So as that plant grows vertically, nodes are regions of meristem that are left behind. And its growth can be inhibited by auxin. So that auxin hormone can actually inhibit growth of the node. And what we see happen, actually, the further the distance the node is from the shoot apical meristem, the lower the concentration of auxin. And then, as a result, less likely an additional growth or an auxiliary bud of the plant is to be inhibited or blocked by the auxin. So as the plant continues to grow, that node gets further and further away from where the meristem, because the plant's growing vertically. Say the meristem's up here. As it continues to grow further away from that node, the amount of oxygen continues to decrease and to decrease. And so auxin can inhibit the growth of the node, but as the meristem grows away from the node, there's less and less auxin, which then allows eventually that node to be able to grow. Cytokines are hormones that are produced in the root, and they also promote auxiliary bud growth. plant tropisms are growth responses to directional external stimuli. And there's two different types that we're going to look at. The first is phototropism, and the second is gravitropism. And the photo is dependent on light. And you've probably seen this before, and you can kind of see it in the image here. This is what happens when the plant grows towards light. You've maybe put a plant on a windowsill, window ledge, and you've seen that plant kind of bend towards the lights. That's what's happening in this case. And then gravitropism is the growth response in response to a gravitational force. And we're going to look at these in a little bit more detail. Auxin can form concentration gradients, meaning that there's different amounts within a gradient. And what this can result in is some changes to the plant. The auxin hormone influence the cell growth rates by changing pattern of gene expression. The auxin hormone gets transported by the PIN3 proteins. And these proteins are delivered to where growth is needed, and in cases, obviously, plants need sunlight, and so there's going to be movement of the plant structure towards light, and as this occurs, it would inhibit the root system from growing further. And so if tropisms in the tip of the plant, so the tip of the stem, detect a greater intensity of light on one side of the stem than the other, auxin is transported laterally from the side with the brighter light to the more shaded side. Here's an image that helps to show this. So this side of the plant is detecting a greater intensity than over here. And so what happens is that auxin gets transported by the PIN3 proteins from this side of the plant laterally to the other side. And so that causes the cells on the shaded side to increase in number, causes more cells to grow, and so then the stem is able to bend towards the light, and then that results in the leaves on the stem to be able to receive more light and photosynthesize at a greater rate. And that's all because of the transport of the auxin from one side to another. Some similar things can happen in the root system, and if the plant has its root on its side, gravity causes organelles to accumulate on the lower side of the cell. And so this allows the PIN3 proteins to direct auxin to be transported to the bottom portion of the cell. And this high concentration of auxin inhibits the root cell elongation. So the top cells elongate at a higher rate than the bottom cells, causing the root to bend downward. And so this is kind of opposite of what we saw in the shoot system with the stem bending. The pattern of auxin effect is opposite in the shoot versus the root. In the shoot, auxin promotes elongation, and the root, it actually inhibits it. So it's the opposite of each other. But it's kind of the same idea with that auxin causing a shift or blocking a shift in bending of the structure. The last topic that we want to look at in this video is called micropropagation of plants. And this is used a lot of times in research purposes. If you're able to identify a plant, a stock plant, a plant that you want to be able to use, this process is basically the extraction of a portion of that plant and the cloning of a portion of that plant in order to be able to produce many, many copies. And this is done in vitro. And it produces a large number of identical plants. You're basically making clones of the plants. And if this process is completed successfully in a sterile environment, it can produce virus-free strains of the plants. And viruses can influence negatively and sometimes positively the plant. This process allows for the plants to be produced virus-free. It's fast and it takes less space than actually growing the plants. So it's quite a bit quicker than just growing the plants traditionally. And so how it works is tissues are sterilized and placed in a sterilized growth medium with plant hormones, auxin, and cytokine to increase cell growth.
Answer the test questions in the online class. Watch the video on the YouTube and send task to the university system. Thank you for your attention.